Assalamu alaikum, everyone. Today, inshallah, we are going to finish this chapter. And the last topic we are going to discuss is how transposable elements might contribute to genome evolution. So we already in the previous lectures saw how crossing over might be mediated by transposable elements. So if this crossing over requires the alignment, the correct alignment between homologous sequences like these. So these transposable elements, they are considered homologous sequences, which means they have identical or very similar sequences, which means the crossing over or recombination can happen between these transposable elements. Now, what happens is that if these transposable elements are misaligned like this, what will happen is that we have incorrect crossing over, and this might lead to gene duplication on one of the chromatids and the deletion of the gene from the other chromatid. Okay? So one of the ways transposable element might contribute to genome evolution is through the incorrect crossing over during prophase 1 of meiosis. Other mechanisms that through which the transposome might contribute to the evolution of the genome involves, for example, the addition of exons. Okay, so imagine if we have this gene, okay, and this gene consists of three exons: exon 1, exon 2, and exon 3. Okay, so this is gene X, for example. And here we have regulatory element or cis element. Cis element means it could be like an enhancer or a DNA sequence that changes basically or controls transcription or the expression of this gene. Okay, and here we have a promoter. Okay, so transposons, they can affect, for example, a transposon might come and introduce a new, let's say, a new, so an exon comes and carries a new exon, okay? Let's call it exon alpha. So this is a transposon carrying an exon sorry, come on. So, okay, comes and insert itself like this between the exons. So now we have exon 1, we have exon 2, we have exon 3, and now the gene also contains exon alpha. Okay. So when this gene is expressed, okay, the mRNA will consist of these four exons. So here, exon 1, this is 2, and this is 3, and here we have exon alpha. So this is an example of a process where a transposon might add an exon to a gene, okay? Also, an exon might insert itself into the promoter of a gene, causing a change in the level of transcription from this gene. So here we have the same gene, okay? And then a transposon with a very strong promoter, okay, might be added like this, so that the expression from this gene increases. So this is a transposon with strong promoter. Okay. So this is another case where here the transposon here did not affect the gene structure per se, but it did affect the level of expression from the gene. Here, there's a change in the gene structure. There's an addition of a new exon, for example. So we could say here, we could call this maybe this process addition of exon, and here we have cis element addition. Okay. Or we have cis element disruption. So a transposon might insert itself. Again, we have the gene. A transposon might insert itself within, let's say, a cis element like this, so that the cis element is no longer able to support the expression of this gene. Okay. So here we have no expression because this cis element was disrupted. Okay, it might be an enhancer, was disrupted by the transposon. So this is the transposon, Tn. Okay. So Tn equals a transposon. So this is cis element disruption. We have also gene disruption. So a transposon might disrupt a gene, might insert itself in the middle of the gene, causing disruption of this gene. So it might insert itself in one of the exons like this. Exon 3, for example, exon 2, then something like this. And here we have the exon disrupting the gene. Okay. So now what is expressed is only the first exon or the first part of the exon. The other exons will not be expressed So this is exon prime, 1 prime, this is exon 3, this is exon 4. So maybe the mRNA that will be produced from this gene will be something like this. Okay. Then we have till the end of the transposon, and maybe there is in the transposon here we have a stop, transcription termination signal. Okay. Instead of having a complete, let's say, a complete mRNA. So this is an example of gene disruption. Sometimes a transposon might cause alternative splicing. So here we have alternative splicing. So insertion of the transposon between exons like this might cause some changes to the way the mRNA from this gene is spliced. So the gene, normally the gene will produce the gene produces an mRNA with the 3 exons like this. This is the normal mRNA exon, not 1, 2, 3. But because of the presence of this transposon, sometimes the presence of the transposon will make the splicing machinery skip one of the exons. So now the mRNA will only consist, for example, of the first. So this is how splicing happening. So we have 1 is joined to 2 and 2 joined to 3. But here 1, because of the presence of this exon, this transposon, 2 will not be included in the final transcript, or 2 will be skipped. Okay. So this is another effect of the exon. So they may carry gene or group of genes to a new position. They also might create new sites for alternative splicing, just like we saw. They might increase or decrease gene expression or protein production, or they might block protein production or make, let's say, disrupt the gene. Also, they might lead to gene silencing. So gene silencing means that we have a gene, let's say, the gene, maybe it's not methylated, or its promoter is not methylated, but a methylated transposon comes in, let's say, or so this transposon is methylated and comes and insert itself, let's say, in the promoter region like this. And because it is methylated here, it will prevent basically the transcription of this gene. Okay. So no transcription. Of course, we have seen how also transposable elements might lead to gene duplication or gene deletion. So all of these are different mechanisms by which transposons might affect gene evolution or protein production. Okay. Another mechanism which I did not mention is basically you have a transposon might carry a gene or a group of genes to a new position. Okay. So here we have an example of that mechanism. You have this gene found in this chromatid, and eventually, because of these transposable elements, this gene is now connected to the other chromatid. Okay. So this is disruption. Sorry. Sometimes I very quick in writing these words. Okay. All right. So this is the end of this chapter. So in this new chapter, chapter 23, we are going to learn about the evolution of alleles, or the change in the allele frequencies within a population. So we're not going to discuss evolution of, let's say, mankind or humans. No, here we're going to talk about the evolution of allele frequency, or the change in allele frequency in a population. And what I mean by population here, I mean a population. This is a group of organisms. Could be humans, could be animals, plants, of organisms. Could be bacteria, any type of organism. Okay. So microevolution. So here we're going to discuss the evolution at the level of the population, not at the level of the individual members of the population. Okay? Individual members, these are the organisms that are part, or they constitute the population. Okay? Part of the population. And you can imagine that a certain population of organisms, like a human, will contain certain types of alleles, and these alleles, okay, or these different copies of genes, each copy of a gene, or each type of a gene, or each allele of a gene will be present in the population at a specific percentage or frequency. And this allele frequency actually changes. How? This is what we're going to learn about in this chapter. How the allele frequencies can change with time, and the three main mechanisms that cause allele frequency change are the natural selection, genetic drift, and gene flow. So first, we're going to discuss natural selection. So again, this is the definition of microevolution: change in allele frequencies. Allele frequency is basically a frequency for an allele is the percentage of an allele among all members أعضاء of a population. Okay. So, for example, we know that blood grouping or blood groups are based on these following alleles. We have IA, IB, and small letter I. So these are alleles. So this is an allele, this is another allele, and this is another allele. Okay, these are alleles. Okay, so if we have, let's say, a population okay, 500, let's say, members or 500 individuals, okay, and the number of individuals, let's say, with, so each individual will have 2 copies of the 2 copies of the gene that is responsible for blood groups, right? And in this case, a 500 members will have 1,000 alleles responsible for blood grouping or blood groups or determining blood groups in, let's say, in this population. And here, the 1,000 allele, maybe we have, let's say, 90 or maybe 100 IA alleles. Okay, we don't care where these alleles are, whether they are basically in a particular or one particular or basically in which members. We don't care. We just we are looking at all alleles within the 15 members أعضاء of this population. Okay, maybe we have 350 IB and the rest are for the allele small letter I. Okay, so now we could say that the allele frequency of I for IA equals 100 over 1,000, which is basically 10%. This is what is meant by allele frequency. Allele frequency for IB is basically 350 over 1,000, which is 35%, and 55% for the allele frequency I, small letter. Okay, so this is what is meant by allele frequency. We look at all alleles. We determine the number of all alleles for a specific gene, for a specific genetic loci or genetic locus. Okay, so there is a genetic locus or a gene responsible for blood groups in humans, and this gene could be of this allele or this allele or this allele. Of course, from these genes we have 2 copies. One copy you inherit from your father, the other from your mother. Now, these two copies could be the same allele or could be different combinations of these three alleles. Okay. So here we have one gene, multiple alleles. Now, within this population, these allele frequencies are not fixed. They might change with time. How? Because of these three mechanisms. And this is how, what we are going to discuss in this course, how these three mechanisms can change the allele frequencies within the population, okay? So something must happen to the population or the members of this population that will cause a change in the allele frequency. All right. So let's first discuss the natural selection. So here we have natural selection, الانتخاب الطبيعي. And we have adaptation or adaptive evolution or تكيف. So one way or one mechanism by which allele frequencies changes is by natural selection. Now, natural selection requires genetic variation to exist within the population, okay? What do I mean by genetic variation? So here, what you see is genetic variation because different individuals or different members within the population, they will have different combinations of alleles. And this is considered genetic variation, which means they will have different blood groups. So different members of the population will have different genotypes for this locus, which means they will have different phenotypes. Now, in order for allele frequency to change and the population, the whole population evolves, there should be some sort of, or there a genetic variation is basically, it's a must in this case. So this genetic variation leads to phenotypic variation. So genetic variation is basically different individuals within the population. They have differences in the genes, in the alleles. Basically, they have different alleles, different genes, different combination of alleles, or any other DNA sequence. This is what is meant by genetic variation. And this genetic variation leads to variation in traits, صفات. Okay. So variation in Arabic, تنوع الجيني, تنوع الصفات, or phenotypic variation. Phenotypic variation. Okay. So genetic variation or the genotype causes the phenotype. A genetic variation causes phenotypic variation. But remember that the phenotype is not only determined by the genes or the genotype. It is also determined by what? What other factors determine the phenotype or contribute to the phenotype or to the final trait? So we have two factors that might affect the phenotype. Any phenotype, genetic factors and what? Anybody? Anyone? Environmental, thank you. So phenotypic variation are determined by genetic variation, but also by the environment, environmental factors. Or environmental variation, let's say, or factors, عوامل. Okay. Now, in order for natural selection to happen and allele frequencies changes, population must have some level of genetic variation. If the population is homogeneous in terms of the genotype, natural selection will not be happening. Okay, or the chance that natural selection will happen will be very low. Okay, so we know that within a population, individuals they have different traits or different phenotypes. And these different phenotypes or different traits reflects genetic variation, or they are caused by genetic variation and in some cases environmental variations. Natural selection can only act on variation, in variation with the genetic component. So natural selection can only happen if there is genetic variation, and it can work on this type of variation, but not the environmental variation. Okay, and we will discuss natural selection in more details, inshallah. All right, so we know that some phenotypes or some traits, بعض الصفات, they are determined by a single gene. Remember Mendel's these plant flower color. Okay, you have, for example, capital P, small p, capital B will produce the same. So these are considered genotype, will produce purple flower color, which is basically the phenotype. And we know that small p gives a white color for the flower. Okay. So now we have one gene with two alleles, allele P capital or allele P small. So this is one gene, one phenotype. So some traits or phenotypes, they are controlled by one gene. But other phenotypes, they are controlled by more genes, by more than one gene, like two or more genes. For example, in humans, skin color, height, طول, and many other traits are controlled by, skin color is controlled by dozens of genes, height, maybe hundreds of genes. Okay? So skin color is basically, it's a phenotype or a trait that is controlled by more than one or two or more genes, or more than one gene. Same applies to height. Now, we can measure the genetic variation within a population or among the members of a population by looking at gene variability or nucleotide variability. Okay. So for gene variability, we can measure gene variability by calculating the average heterozygosity. So heterozygosity means, so this is, for example, these genotypes, this genotype is considered homozygous. An example of a homozygous genotype, because both alleles are the same. Here we have also this genotype is also homozygous. But here we have two different alleles. This is heterozygous genotype. Okay. So heterozygosity, we are talking about the two copies of a gene, or the two alleles of a gene are different. They have different sequences. Now, we know that here loci could mean, here we could say that loci as a gene or DNA segment. DNA segment with sequence variations among individuals. Okay. So if we talk about, for example, we have around 21,000 genes, okay, or let's call it protein coding genes in humans. Okay, each one of these is considered a locus, and these are actually equals 21,000 protein coding loci. We could call them also protein coding loci. Loci is the plural of locus. Loci mawaka. Here, locus mawaka. Okay. So when we talk about loci, we mean genes or maybe DNA fragments or DNA sequences that are different among different individuals. There are some differences in their sequences. Now, the average heterozygosity, we want to go into a population and determine how many members of the population, how many members of a certain population, they will have heterozygous alleles, or they have heterozygous genotypes at, at the different loci present in their genomes. Okay, the more heterozygosity observed among members of the population across all loci, the higher the gene variability. Okay. The higher the gene variability observed in a population, the higher the genetic variation among within this population. Okay. So one leads to the other. In terms of nucleotide variability, here we're talking about so gene variability. Here we're talking about differences at the allele level. So different alleles, okay? If different genes within the population, or a high number of genes within the population, there are two different alleles for each gene within the population. This means the average heterozygosity will be higher, which means the gene variability will be higher, which means the genetic variation will be higher. Now, nucleotide variability here, we're comparing DNA sequences. So we are looking at the differences at the level of the nucleotide sequence. Okay. Sometimes a nucleotide variation might not lead to a genetic variation, which might not result in a phenotypic variation. Why? Because nucleotide variation does not, so differences in nucleotide sequences might not produce different alleles, because most differences, or most, nucleotide sequence differences, they happen either within introns, as you can see here. So this is an example of a gene consisting of one exon, 1, 2, 3. Okay, so these are the exons of this gene. So this is exon 1, for example. This is exon 2. This is 3. This is also 3. This is part of 3. And here 4. So this is a gene consisting of 4 exons. And we have these introns. So this is intron 1, intron 2, intron 3. Okay. And as you can see, these orange lines, they represent nucleotide variations. They could be base pair substitutions. So these are locations where different individuals or different members within a population, they have different nucleotides. Okay. And these variations, nucleotide variation, could also include some deletions or some insertions. And as you can see, because introns usually they are longer than, let's say, exons, most of these variations might happen within non-coding sequences like introns or outside the gene. Okay. But sometimes, even if they happen within exons, there might be, silent, let's say silent, they might not change the amino acid. They might not cause a change in the amino acid. Or they might cause a change in the amino acid from one type into the same type. So here in this example, we have almost 43 nucleotide sites with variable base pair. Okay. So here we have these. If you count these lines, these lines represent nucleotides that are different among different individuals within the population. But of these 43 base pair substitution, only one result in amino acid change. The other ones, they do not result in amino acid change because some of them are in introns, in non-coding sequences. Okay. Even the ones that within coding sequences, they do not change the amino acid, or they do not change the codons, or they do change the codon, but they do not change the amino acid sequence of the protein encoded by this gene. Okay. Only one site, for example, this one, only this nucleotide, when it is substituted, will cause a change in the amino acid sequence, which might cause a phenotypic variation. So this is a nucleotide variability here. Okay, so this is a nucleotide variability. And here also we have nucleotide variability. Okay, each one of these is considered nucleotide variability. But this nucleotide variability causes a change in the amino acid, which means it causes gene variability, because this nucleotide variability actually results in a change in the codon and the amino acid encoded by that codon. Okay, and this gene variability leads to genetic variation, and so genetic variation, which leads to phenotypic variation. This is another example of nucleotide variability, but this nucleotide variability does not cause any gene variability, which means it does not cause any phenotypic variation. Same applies to this one, this one, this one, and many other ones that are found here. So this is the only one. Why? We know because some of these substitutions, they will cause a silent mutations, so the amino acid does not change because of the redundancy in the genetic code, or they might just convert one amino acid into another type of amino acid of the same type. So there will be no change in the amino acid sequence or in the properties of the protein. And as a result, there will be no change in the phenotype that will result from the function of that protein. Okay. Okay, so before we go and discuss what causes what are the sources of genetic variation, let me again point out that phenotypic variations among individuals in a population is the result of the genetic variation, but also it's also a result of the environmental factors or environmental variation. So both environmental and genetic factors, they determine the phenotypes observed in individuals in a population. Okay. So now next we're gonna look at how genetic variation, okay, occur or happen in the populations. Okay. So the origin of genetic variation is basically the mutations or gene duplication. So new genes or new alleles that will lead to genetic variation, they are generated by mutations or gene duplication. Okay. Also, we know that sexual reproduction can result in a genetic variation by recombining different alleles, but these alleles already found in the population. But a population might have basically new genes introduced into its gene pool or new alleles by mutations or gene duplication. Okay. So the first thing we're going to discuss, the first mechanism of that increased genetic variation is the formation of new alleles or generation new alleles, and this could happen by mutations. So mutations, they might generate, they will cause a change in the nucleotide sequence, and this change might lead to new alleles, new gene sequences and new alleles. Okay. Now, if these mutations happen in the cell lines, in the germ lines, so germ lines are the cells that are responsible for producing gametes, sperms and eggs. So gametes and eggs are produced from diploid cells. And if mutations affect these cells, this means that these mutations and the new alleles generated by these mutations could be transmitted from one generation into another. Okay. Now, one type of mutation is called point mutation, which is basically a result in a change in a single nucleotide. This point mutation might have an effect on the phenotype, and this effect might be harmful. Harmful means مؤذي, سيء. Okay? And some mutations are bad, or they have a harmful effect on the phenotype, but some other mutations might be beneficial, مفيدة. And this might lead to a new beneficial allele. Okay, but these mutations are rare. So most of the mutations, they will affect the phenotype of the gene negatively. They are harmful. And some of these mutations, these harmful mutations, might exist within the population, hidden in recessive alleles. Okay? So remember, some alleles are dominant, some alleles are recessive. If the dominant allele does not contain harmful mutations, the phenotype will depend on the dominant allele. But a recessive allele containing harmful mutation could still exist hidden, because the phenotype is not affected by the presence of the recessive allele, or does not depend on the presence of the recessive allele. Okay. Typically, natural selection will select against these harmful mutations, but these harmful mutations might remain within the population, hidden in recessive alleles. Okay, so they would be hidden from natural selection in recessive alleles. Now you have to understand, all the alleles that are stably found in a population, okay, so if we go back to this population, if we are talking about, let's say, these three alleles that are responsible for blood groups, these alleles are found in a population. Now, natural selection already, okay, so natural selection already selected these alleles. Okay, so because the mutations might, like a mutation might generate an allele like this, but if this allele is harmful, natural selection will remove this allele from the population. How? If an individual has this allele he or she might die, and if they die and they cannot reproduce organisms or offsprings or children, this means we are removing this allele from the population. Okay? So any individual with this harmful allele will die before, maybe at birth, or before they can get married and distribute this harmful allele into other members of the population or into the next generation of the population. Okay? So the organisms within the population, their alleles already went through the natural selection. And this natural selection ensures that these alleles within the population are well matched with the environment surrounding the population. So here we have a good fit or good matching between the alleles found in the population and the environment surrounding these individuals. So members of this population, they are able to survive their surrounding environment because they have alleles that allows them to do so. Okay. So the idea here is that you have an environment. This is a very important bia. And you have a population living in this. Okay, these are all, let's say, this is a population living in the environment. And there are alleles found in this population or among the members of this population. And these alleles, they fit or allows the members of this population to survive the environment. Okay? So natural selection will select the alleles that and hence the phenotypes that will allow members of the population to be in a good match with their environment. Okay? So basically, the genes we have in our genomes, the alleles we have in our genomes, they have been selected, and this is why we can live in the environment surrounding us. Okay? Some mutations might affect non-coding regions, and these are neutral variation. They cause neutral variation. So basically, they do not offer any selective advantage or disadvantage because they do not affect coding regions. Okay? And some mutations to genes, they might be neutral because of redundancy in the genetic code. Remember, more than one amino acid more than one codon can encode for the same amino acid. So for example, if we, so for example, you have these 4 codons encode for the same amino acid. Okay. Let me just look at some codons. Where's the codon table? So you have CUU, CUA, CUC, and CUG. These all codons, they encode the amino acid leucine. So this is what is meant by redundancy, tikrar. Okay, so if you have a codon within a gene that is CCU and a mutation hits the third nucleotide of this codon, and now you have CU, for example, A, it doesn't really matter because this codon encodes leucine and this mutant codon also encodes, use different colors, encodes the same amino acid. Okay. Okay, so genetic variation can be caused by the introduction of new alleles or new genes. And these new alleles or new genes might be generated by mutations. Another mechanism by which genetic variation increases is by changing in the gene number or position. Okay, here, when we talk about gene number, we're talking about the number of genes per haploid set inherited from one of the parents. So some chromosomal mutations or large scale, so here we're talking about large scale. Remember, we talked about large scale and small scale mutations. Large scale mutations, these are changes that happen at the level of the chromosome as a whole, and they affect large fragments or large segments of chromosome. These large scale mutations could lead to the deletion, disruption, or rearrangement of many loci. And if these loci, they contain genes, this might lead to genetic variation. Some regions of DNA might be duplicated, as we saw in the previous chapter. And these duplicated genes might accumulate mutations, and now they become new genes with the new functions. Okay, and we looked at example how the globin alpha and beta globin gene families, they originated from one ancestral gene that underwent duplication, mutations, and transposition. So gene alteration in gene number or change in gene number and position might happen because of one of three, let's say, mechanisms. So we know, duplication. I'm talking about duplication. Duplication might happen because of errors during meiosis. For example, unequal crossing over because of misalignment between transposable elements, or it could happen directly because of transposable elements and because of slippage during DNA replication. So we have large fragments of chromosomal segments are duplicated, and if these chromosomal segments that are duplicated, they contain genes, this means we have duplication of genes. Okay. Rapid reproduction also increases genetic variation. So In plants and animals, the mutation rates are very low compared to other organisms. Okay, so on average, there is only one mutation in every 100,000 genes for every generation. Okay. There are even lower mutation rates in prokaryotes, but prokaryotes, they reproduce more rapidly than animals and plants. Viruses, they have high mutation rates and they have short generation times. And this is why viruses, they keep changing their genetic material and they keep changing. And that's why we don't have some treatments for certain viruses. So we cannot treat influenza viruses. And now we are currently having problems with the coronavirus because the coronavirus genetic material keeps mutating. Okay, the mutation rates especially if the virus contains RNA genetic material, not DNA. Okay. So in some organisms, the rapid reproduction, like in prokaryotes and viruses, leads to or accelerates or increases the genetic variation. But in animals or in organisms that reproduce sexually, genetic variation might result because of the recombination of alleles, because of shuffling of alleles. Shuffling means خربط. You have a sperm carrying certain types of alleles, okay? And you have an egg carrying different types of alleles for different genes, let's say. And then these two cells, when they mix together, they will form a zygote with, let's say, a new combination of alleles that already found in the population, but this is a new combination of alleles. We're talking about maybe a new combination of alleles. Okay. So in sexual reproduction, تكاثر الجنسي, alleles can be shuffled into new combinations. So existing alleles. So we're not talking about generation of new alleles. Here we have these alleles that are found within the population, and these alleles could be the source of basically combining these alleles from the sperm and from the egg could result in a new combination of allele and increases the genetic variation. We have three mechanisms that causes shuffling of existing alleles. We have crossing over, because the crossing over in prophase 1 of meiosis 1 generates new recombinant alleles. We have independent assortment and fertilization. Okay. Both crossing over and independent assortment, they happen during meiosis. And fertilization, this is when a sperm fertilizes an egg. All these three independent mechanisms lead to shuffling of already existing alleles in the population, which leads to basically an increase in genetic variation.